Okay, so the topic of this video is going to be mutations, DNA misspellings, DNA errors. And that's what mutations are. Let's go ahead and get started. As we go through this video, you're going to want to have a copy of the genetic code with you, whether you have it in your textbook or whether you Google the words genetic code and print out a diagram like this. It's going to be really helpful to have this alongside of you as we go through this video. I'm also going to assume that you have a general understanding of how transcription and translation works, because you're going to see transcription and translation discussed in this video. So if you're not quite sure what the process of transcription and translation are, you may want to watch those videos first before you try to tackle this video. So let's get started. Okay, so I first want to discuss a point mutation. Well, in the animation, what you see happening is the process of transcription. The black letters across the top are DNA, the red letters that were created are messenger RNA, and so transcription just occurred. And what I'm going to do is I'm going to underline, starting with the letters AUG. If you remember, AUG is the start codon. And I'm going to underline every three letters, every three nucleotides. Again, I'm going to underline what are called codons. And the ribosome will read the codon AUG, and transfer RNA will deliver the amino acid called methionine. The ribosome will read the codon UCC. Look up UCC on your genetic code chart. And transfer RNA will deliver serine. The ribosome will read GAA. Look up GAA on your chart. Transfer RNA will deliver glutamic acid. The ribosome will read the codon AGC. Transfer RNA will deliver serine. The ribosome will read the next codon, CCG. Transfer RNA will deliver proline, and then the ribosome comes to UGA, and that's a stop codon right there. So this was a review of transcription and translation. Now what I want to mention now is what are point mutations. A point mutation is where one nucleotide is substituted for another. So at the bottom are the same strip of black DNA letters. And notice how I've just changed one of the letters, one of the nucleotides. You can see in the letters on top, originally that T that's pulsing originally was an A. So we're going to pretend that in this mutation, the T that's pulsing and flashing, that's a mistake. It's not supposed to be a T. It's supposed to be an A. So we have a misspelling in our DNA. One thing I want to mention is that misspellings or errors or mutations like this are often repaired by spell checker enzymes, but what I want to show you today is what happens when they're not. This might lead to a change in amino acids, which could have a drastic impact on the overall protein. So through the process of transcription, the red letters are forming, that's messenger RNA is being formed. The messenger RNA will break away and move to a ribosome, and I'm going to underline, starting with AUG, I'm going to underline every three nucleotides. And so the ribosome will read the codon AUG, transfer RNA will deliver methionine, just like we see in above. But now we come to the mutated codon. Above, in the top of the picture, the codon is supposed to be UCC, for the amino acid serine. But because of that misspelling in the DNA, it's not UCC anymore. It's now ACC. Transfer RNA is not going to deliver serine in this example. It's going to deliver threonine. The ribosome goes to the third codon. Well, the GAA codon is the same. That hasn't been mutated, so that's still glutamic acid. The fourth codon is the same. It hasn't been mutated. That's still serine. And the fifth codon is the same. That hasn't changed. That's still proline. And then we come to codon number 6, and that's still the stop codon. So that one change, it's supposed to be serine on top, but you see, because of that mistake, it's threonine. I'm going to explain why that's a big deal in just a moment. But before I do explain why that's a big deal, that one changed amino acid, I want to show what's called a silent mutation first. Here's again a strip of black DNA letters, and here's our misspelling. Here's our mutation. You can see it's the flashing letter A. It's not supposed to be an A. In the strip above on top, you can see it's supposed to be a G, but because of this misspelling, this mutation, it's now an A for adenine. Through the process of transcription, messenger RNA is going to be created. Notice how the messenger RNA is now also misspelled. It's flashing in red. The red U, that's a misspelling there. Again, the messenger RNA was using the DNA as a template. So the red mRNA goes to a ribosome. I'm going to underline every 3 letters. And the ribosome reads the codon AUG. That hasn't changed. It's still methionine. But now we come to the codon with the flashing letter U. On top, you can see it was supposed to be UCC for the amino acid serine. But because of the misspelling, it's not UCC anymore. It's UCU, which if you look on your genetic code chart, it's still serine. This is what we mean by a silent mutation. There's a clear misspelling, but it's silent in that there's no impact. The person got lucky because the amino acid has not changed. UCC and UCU, they are both codons for the amino acid serine. So you go to the third codon, GAA, that's glutamic acid, nothing changed. The fourth codon is the same, that's the codon for serine. The fifth codon is the same, that hasn't changed, it's proline. And then we come to the sixth codon, that's our stop codon. So this person has a clear misspelling, a clear DNA mutation. But this is what we mean by a silent mutation. It didn't cause any impact, any change to their proteins. Okay, now I want to go back and explain why just one wrong amino acid can be a really big deal and have drastic side effects. You may have heard of the protein called hemoglobin. Well, hemoglobin is a protein found in red blood cells. It helps red blood cells carry oxygen around your body. Hemoglobin is a protein, and like all proteins, are made from a bunch of amino acids. In fact, hemoglobin is made from 574 amino acids. Now, if you've heard of sickle cell disease, sickle cell is caused when people with this disease, one amino acid has been changed in that entire strip of 574. And because of one wrong amino acid, they can have a life-threatening disorder. Let me show you how this works. The letters in top are DNA letters, and through the process of transcription, we'll make these letters beneath it of messenger RNA. The messenger RNA will go to a ribosome, and the ribosome will read GUU, and that's the code for val, which stands for valine. The ribosome reads CAU, and that's histidine. The next one in blue is leucine, in orange it's threonine, in pink it's proline, and the last two in that kind of yellow color are glutamic acid. So this is the normal coding for people who have normal hemoglobin. And if your hemoglobin's normal, your red blood cells will look like this. Round, perfectly, I shouldn't say perfectly round, but nice round red blood cells carrying oxygen all around your body. May you live happily ever after. But now, here's where people have sickle cell disease. Look at this. There's a misspelling. I've highlighted it with the red letter A. You can see in the normal cell, the code is supposed to be CTT. But in sickle cell, people have the code CAT. So through the process of transcription, when they make messenger RNA, you can see the messenger RNA is now GUA. In the normal cell above, the messenger RNA is supposed to be GAA. But because of the misspelling, it's now GUA. So through the process of translation, the transfer RNA is going to bring over, in this case, the wrong amino acid. You can see where we get to that, the 1, 2, 3, 4, 5, 6. We get to the sixth codon, the GUA. That is, if you look up GUA on the chart, that's the code for valine. That's the wrong amino acid. You can see the normal cell on top, there's supposed to be 2 glutamic acids back to back. GLU, GLU, back to back. But people with sickle cell, one of the amino acids has been changed. It's been changed to valine. And because of that one change, their red blood cells look like this. Banana-shaped or sickle-shaped. If you've ever seen a sickle, it's like a blade with a curvature to it. And so because of this, people have a life-threatening disorder called sickle cell disease. 574 amino acids, and one of them is wrong. So this process of assembling amino acids has to be perfect. This is a great example of what can go wrong if just one amino acid has been changed. The next type of mutation I want to mention is what's called a frame shift mutation. It started off the same way. We have red messenger RNA being created through transcription, and I'm going to have the ribosome read every 3 letters at a time. So starting with AUG, that's the code for methionine. UCC, that's the code for serine. GAA, that's the code for glutamic acid. AGC, that's the code for serine. CCG, that's the code for proline. And then UGA is a stop codon. Well, what a frame shift mutation is, it's where a nucleotide gets inserted or accidentally deleted from an entire strip of DNA. And so the entire sequence after the DNA or the RNA is mutated is going to be shifted and altered. And you'll see what I mean by this in a moment with my animation. So on the bottom, here's an example of a frame shift mutation where a letter is going to be deleted. So here's the same letters in black, the same DNA letters in black that you see. Notice how I have the T flashing. The reason for that T flashing, watch what happens. Accidentally, that T for thymine is going to be deleted. Notice all the other letters after shifted down. So why is this going to make a big deal, big difference? Well, through the process of transcription, the red messenger RNA is going to be created. And again, I'm going to underline, starting with AUG, I'm going to underline every 3 nucleotides. That's called a codon. And the red mRNA goes to a ribosome. And AUG, when it's read by the ribosome, that's the amino acid methionine. UCC is serine. GAA is glutamic acid. So far, nothing's changed. If you look at the top picture, look at the bottom picture. They're identical. But here's where all the changes start to happen. Because of the removal of that T, the letters have completely shifted. So now we have GCC for the code for alanine. The next codon, CGU, is for arginine. The next codon was supposed to be a stop codon. There was supposed to be a stop codon, a UGA stop codon, but because of the shift, the next codon is now GAC for aspartic acid. The next codon is now histidine. The stop codon has been basically destroyed. So this ribosome doesn't know to stop creating the protein, stop assembling amino acids. So ultimately the ribosome just keeps on going, and eventually it runs out of messenger RNA to read, and the protein that's created you might even call a nonsense protein, because it's going to be very, very dysfunctional, if any function whatsoever. So here's an example of a frame shift insertion. Kind of the same result, but instead of deleting a letter, we're going to insert a letter. Watch this. Here comes T for thymine that just got accidentally inserted. I'm going to have it blink the whole time so we can keep our eye on it. So that 4th T is not supposed to be there. In the picture on top, you can see there's supposed to be 3 T's in a row, TTT. But because of that insertion, it's now TTTT. There's 4 T's. So through the process of transcription, red messenger RNA is going to be created. And notice what happens is that that 4th T will lead to the creation of a 4th A. Well, the messenger RNA will go to the ribosome, and I'm going to underline every 3 letters at a time, codons. And the ribosome will read AUG, that's methionine. The ribosome will read UCC, that's serine. So far, everything's fine. The ribosome reads GAA, that's glutamic acid. So far, we're fine. But now here's where the shift occurred. Because of inserting that T where it doesn't belong, everything after the T was shifted. And so now the codon is AAG for lysine. The next codon is CCC for proline. Again, we're supposed to have a stop codon next. The stop codon was supposed to be UGA, but because of the shift, because of the addition, the insertion of that extra T, that extra nucleotide, it basically ruined and destroyed the stop codon. It's now GUG for valine. The next codon is now ACC for threonine. The next codon is AUU for isoleucine. The ribosome didn't know to stop because the stop codon was basically destroyed. So this protein that would be created, again, would pretty much be a nonsense, non-functional protein. And so, as I just mentioned, stop codons are often lost as a result of a frame shift mutation. And the proteins that are created, I've said this a few times, are often non-functional due to the large number of incorrect amino acids. Maybe the protein is just too big now. It's only supposed to be made from maybe 5 amino acids, but now it's made from 20 amino acids. So frame shift mutations are a lot more serious. Well, what about a chromosome mutation? The frame shift and the point mutations, these are mutations that happen to individual genes. What about a chromosome mutation? Well, there's a process called translocation. And this is where chromosome segments will combine with non-homologous chromosomes. So I have 2 non-homologous chromosomes, chromosome 5 in blue, chromosome 8, which is a little smaller in red, or excuse me, chromosome 8 in green. And what happens during translocation is that genes of one chromosome end up on entirely completely different chromosomes. Watch this. Through the process of translocation, a portion of chromosome 5 may actually break off and combine to chromosome number 8. So now you have genes that are supposed to be on chromosome 5 now attached to chromosome number 8. And often what happens in this process is that an entire gene might be destroyed. For instance, the AUG start code might be on chromosome 5, but the UGA stop code now might be on chromosome number 8. So the gene was literally ripped apart with half of the gene on chromosome 5 and half of the gene on chromosome number 8. So you can imagine if that's the case, the gene, if it was cut apart, the gene is not going to be a functional gene anymore. The person's not going to have the benefit of this protein that they're supposed to have. So a few chapters ago, we discussed karyotypes, pictures of a person's chromosomes. Well, here's a karyotype of a person with Down syndrome. Now, we've seen Down syndrome karyotypes before. They have a person with Down syndrome, whether it's a male or female, has 3 chromosome 21s. Well, when you look at this and you find chromosome 21, I only see 2 of them. But if you look harder, there's a 3rd chromosome 21. There's 1 chromosome 21 right there on the left. There's a 2nd chromosome 21 right there. The 3rd chromosome is part of has been translocated onto chromosome 14. So this person still has Down syndrome, even though it may not look like it. They still have 3 chromosome 21s. It's just one of the chromosome 21s has been translocated onto and attached to chromosome 14. So there's a great example of translocation from a moment ago. So what impact would this have on offspring? Well, first of all, it depends on what kind of mutations we're looking at. Are mutations, are they in somatic cells? Remember what somatic cells are? Skin cells, nerve cells, muscle cells, blood cells. Somatic cells are body cells. Somatic cells are non-sex cells. And so these mutations only affect the individual. Here we see a person with a pretty nasty sunburn. Well, sunburns can eventually, over time, if you have repeated and serious enough of sunburns, can cause skin cancer. Mutations can occur and can cause you to develop skin cancer. Now, pretend that this guy right here, our cartoon guy, due to years of sunbathing, this male developed skin cancer. His wife recently gave birth to this boy that we see here. Will this child also inherit the skin cancer mutation that the father has? I hope you see the answer to this question would be no. In this case, again, the father developed skin cancer maybe because of his lifestyle of sunbathing. So if a mutation is only in a somatic cell, well, somatic cell DNA is not passed on to future generations. You don't make a child from DNA of somatic cells. You make a child from DNA of the gametes, the sperm and the egg. So in order for a mutation to be passed on from parent to child, that mutation has to be in the sperm or the egg. So if we look at what germ cells are again, and germ cells aren't what you think they are. Again, when we hear the word germ, we think of gross, viruses and bacteria and we got to wash our hands. But that's not what germ cells are. Earlier in the school year, we talked about germ cells. They are the diploid cells that will undergo meiosis for males to make sperm and for females to make eggs. So in the male reproductive system, there are germ cells that will go through meiosis to make sperm, and in the female reproductive system, there are germ cells that will go through meiosis to make the eggs or the ovum. And so in my animation here we have 7 or 8 or 9 sperm cells, and I have one of them with an M inside because we're going to pretend that this sperm cell with an M that's also flashing and pulsing. But the M stands for it has a mutation in its DNA. The other sperm cells are fine, but this sperm cell pretend has a mutation. Now, if this sperm cell with the mutation were to fertilize the egg, which would then become a zygote and develop into a fetus and then a baby, eventually this person that is born, whether it's a male or female, they very well could inherit this mutation because the mutation was in a sperm cell in this case. Of course, it could work the other way around. Maybe mom's ovum had a mutation, and of course that could be passed on to the child as well. But the point I want to mention is that mutations are only passed on if there's a mutation to a sperm or an egg. If the germ cells that make the sperm and egg have a mutation, then those sperm and egg very well could be mutated, which could lead to the child inheriting some sort of DNA error. So what causes these mutations? Well, let's try to avoid them. Well, it's not entirely easy to avoid some of these things that cause mutations. Well, first of all, just general objects that cause mutations are what we call mutagens. And so the reason for this picture, I love this picture. This is a picture of our sun giving off UV light. And I bring up UV light because UV energy, UV radiation is a known mutagen. It's a known mutation causing object, or energy I should say. And so what often happens is mutagens, they speed up the replication process. The process of DNA making a copy of itself is often sped up because of mutagens, which could increase the chance for error. And mutagens also cause nucleotides to break apart. And when they break apart, they often will recombine in wrong patterns. And so the animation shows here this sunlight. Sunlight is being absorbed into our skin. Let's say you're at the ocean and you're enjoying a day at the beach and you forgot to put on sunscreen, or you're not wearing a long shirt and you're not underneath an umbrella and you've been exposed to sun for a long time. And watch what happens. When your skin absorbs UV rays, what it causes is your DNA molecules to break apart. The A's and the T's separate from one another, and so UV radiation is a known what is called a mutagen. And so of course you want to limit your exposure to mutagens such as UV. So if you are going to be at the beach, of course wear sunscreen, and not just once when you get there. You may be at the beach all day. You may spend 10 hours at the beach. So you have to reapply, of course, the sunscreen according to the directions on the bottle. Of course, there's other protections you could take, wearing lip balm and wearing sunglasses and wearing a hat and sitting underneath an umbrella and a light long sleeve shirt. These are things that you can do to limit your exposure to UV, which again is a known mutagen. So there you go. If you're in my biology class, hit the pause video and try to answer these questions. And I'd love to check your answers for accuracy, either before school or after school one day. Good luck.